Ace Podcast.
Hello everyone and welcome back to Vaguely Accurate, the science show where we meet, learn and discuss with early career and higher degree researchers. Today's show format is a little different. I was invited to present and speak at the Research Bazaar conference at Curtin University recently. And while I was there, I did 4 short brief interviews to showcase some of the attendees of this workshop. So that's what today's episode is. 4 short interviews lasting approximately 5 minutes each, challenging the interviewee to concisely convey their research to a lay audience. And just to give you a bit of background about the Research Bazaar conference and workshop, it's designed to improve the computer literacy skills, such as coding, of all attendees. There are a few electives where guest speakers, such as myself, come to talk about specialist subjects such as science communication and the opportunities that are out there. Having the opportunity to present and attend to this conference and workshop at Curtin University, I'd like to thank Curtin and all the organisers that made this possible. So without delaying us any further, I'd now like to lead us into the 4 interviews aptly mentioned. Cheers, guys.
Yeah, so I'm Sherry Yan and I'm from Murdoch University. My field is in neonatal research, so that's bacterial infections in babies.
That's really interesting.
Yeah, it is. So I get to work with babies and clinical staff, and it's interesting because I get to do recruitment. So I start up the study, the clinical study, and then I recruit the babies, and I get the samples, I process it. So I'm basically from ground zero upwards.
That's fantastic. Yeah. Maybe a silly question, but how does a baby volunteer for scientific experimentation?
Yeah, so basically we get consent from the parents, and that's how we get the babies into our study. Yeah.
So do you mind telling us briefly, what is your research exactly? What are you looking into?
So my research is trying to find a better way to diagnose bacterial infections in babies. Okay. So that's mostly what my research will be doing.
What motivated you to study this?
I think my interest in undergrad was mainly in bacterial infections, and I was also really interested in child health. Yep. So that led me to where I am now in finding a cure or finding treatments or better ways to diagnose bacterial infections in child health area. Yeah. But I also believe that not much advertisement or marketing is put into funding for child health studies. So we hear a lot about cancer and Alzheimer's and a lot of the adult studies, but we don't hear as much about children studies as
So what would be the application of your research? Are you looking at any particular bacterial diseases in particular that some people may or may not have heard of?
No, we actually look at just all bacteria that induces infection in the babies. So what we want to do is find a diagnostic test that can just detect bacterial infections in babies so that they don't preemptively be treated with antibiotics, which can cause antibiotic resistance, which of course we know is a global problem. So we try and nip it in the bud
really early.
Yeah, really early. So.
And second from last question I want to ask is, do you look into anything with the gut bacteria as well, or is it you just looking at more the infectious, dangerous side of the bacteria?
So for my research, I'm focusing on the blood-borne bacteria, so not so much on the gut. Okay. But I know of other people who are doing research in the gut microbiome, which is also very important nowadays. Yeah. So yeah.
And lastly, what brings you to Research Bazaar? What have you hoped to gain from being here?
So the main reason I'm here is because a lot of the data skills that I require will be in command line, and a lot of the softwares and programs that they offer here is what I will be using for my data analysis. So it's a good introduction and an intensive way to learn what I need to learn in a short period of time.
Awesome. Thank you very much for talking to me.
No worries.
Yep. Hi, I'm Laura, and I study the ecology and biology of carnivorous plants.
And what is it that you're actually studying about, and what is your PhD on?
The basic question is, how hungry are they? So we want to know basically how much do these different types of carnivorous plants rely on capturing and digesting insect prey to get the nutrients they need.
And this is just a question that I need to know.
Yeah.
When you say how hungry, what do they eat? Because carnivorous plants, I'm still imagining rainforest, big
Yeah,
man-eating fly traps.
Luckily they don't eat any humans, so I've been fine when I go out on field trips.
That's a big save.
Yeah. Usually it's small insects. Different types will catch different things. So you have climbing Drosera, for example, which will catch lots of flying insects like flies. And then you've also got ones that lay flat on the ground, and they might catch more of the smaller crawling insects. A lot of the bigger insects like bees, for example, would be a bit too strong sometimes to actually be caught by these plants.
So what's the motivation that made you kind of move into the ecological botany field?
Well, I was always interested in nature and the environment, and I love David Attenborough. So
who doesn't?
Yeah.
Absolutely legend.
Yeah. And really I just find the plants of southwest Australia to be really fascinating because they have all these interesting ways to get nutrients. Our soil is so infertile, so poor in nutrients that plants have just had to find other weird ways other than just using normal roots.
I spoke to someone recently and they were telling me how plants and fungi have a symbiotic relationship in order to get these. They kind of work symbiotically to extract nutrients from the soil. Is that what you're referring to in some respect? Well, it's one mechanism.
That is, so that's one mechanism. That's like mycorrhizal fungi, and then you've got also carnivorous plants, which I study. There's also parasitic plants, like the Western Australian Christmas tree, which...
Of course your Christmas trees are carnivorous.
Yeah, or parasitic. But yeah, it's got these kind of roots which run along and clamp onto another root and then cut in and steal the nutrients.
Kind of like a wood wide web.
Yeah, that's right.
So what would be the significance or the application of your research? Even if it's at the early stages, do you have any hypotheses for what you hope to come out of what you study?
Well, basically there just really needs to be more research into carnivorous plants, in my opinion, especially in Western Australia because we have more here than anywhere else in the world. So a lot of the carnivorous plant research has focused on other areas of the world. But one of the problems for carnivorous plants is that they are threatened by habitat change and land degradation and even poaching. People that go out there and take plants from the wild to have at home or to sell on eBay, things like that. So really, with my research, I want to raise awareness about these issues and also just better understand the biology of these plants and what they require in their habitats so that we can conserve them into the future.
If you're looking to raise awareness, would you be keen to come onto a full-length feature show one day or sometime soon and talk about your work?
Yeah, I'd be happy to.
Fantastic. Yeah. And last question, why are you here at RezBaz? What skills do you hope to develop in the workshops?
Well, I'm here at RezBaz because I've never really felt confident with R as a sort of coding. It's always seemed a little bit confusing and I would always get error messages and not know why.
As far as I'm aware, everyone, no matter how experienced, she's still going to get error messages.
Yeah.
Don't take it personally.
Yeah, so I really just want to learn more about that, but I'd also love to learn more about other ways of communicating research, other digital tools that can be used other than just Microsoft PowerPoint, which I do love, but I'm sure that there are other really cool options. Yeah, exactly. Yeah.
Like podcasting.
Hey,
like us.
That's right. Yeah.
Brilliant. Thank you very much for talking to me.
That's all right.
I'm Maxim, and I'm on an international student visa in Australia to do my PhD, and my studies in applied bioinformatics. In my case, I'm studying plants, in particular narrow-leaf lupin, which is a legume plant.
Okay.
And so applied bioinformatics is sort of, I call it understanding biology using computational sciences.
Understanding biology through computational sciences.
Yeah, sort of, because what we do in this field, we get a lot of sequence data for our plants, in my case, and we try to somehow decode what these sequences means in terms of what genes are available there and all lots of different type of a story.
Bioinformatics and computational science is an incredibly growing field at the moment. Like, it's almost, you all have to have that specialty or understanding in order to do anything, to really understand. It's more, rather than answering small questions, it's mass data and then working correlations out through the data.
Yes, sort of.
So what's your motivations to move into that field?
I've done a lot of research before deciding to go to that particular field, and I think the main motivation was the demand for the field, because it's quite sort of new and you need to have the biology understanding, and also you need to have the computational sort of skills, which is not easy because if people are good in biology, usually it means that, okay, they are possibly not very, very good at computer sciences.
Yeah.
So it sort of needs different type of skills, and not many people are willing to stretch their capability to go through that path. But, okay, that's what makes it demanding.
Where did you learn the skills? Did you learn them at university?
I did like a self-study.
Okay,
Yeah, mainly through online courses, and I got lucky with a bioinformatics group moving to UWA. So I've got lucky with nice people in the group who somehow walked me through some steps. Because anyway, when you start, you need some sort of, at least some steps, how to start, and because there are lots of packages out there. You need sort of knowing.
You need that foundation to get into the field.
Yes, that's true. Yeah.
So what is the application or the significance of what you do? So when you actually...
So, I don't know how much you know about sequencing and...
I understand a basic now.
I think the main example would be like the human sequence, which everyone knows about, and it sounds very fascinating. But we do the similar type of thing on plants, with the hope that if we could unlock the genome of the plants in terms of what exactly these sequences means, and we find which genes, for example, are present in wild exogens of a plant which are absent in domesticated type, then it could help potentially if we recognize these differences between the population or, different studies, then we potentially could put back the genes into our domesticated crops in order to having a better crops, like super crops, sort of.
So it's like the next step of genetic modification for crops.
That would be some sort of a first step of recognizing things, and if you get lucky to find something interesting, then you need to give it to people who works in practice, in the field and in the laboratory to put genes back somewhere. Yeah.
That's really interesting.
It is.
And final question: why are you here at RezBaz? What language are you hoping to learn?
I'm mainly registered as a helper.
Yep.
But then I got that. Apparently they have quite a few helpers, and then I found this top of a Python course using Jupyter Notebook, which I never used myself, and I find it quite interesting. So I just turned into being an audience right now.
Awesome. Thank you very much.
Thank you.
So I'm Paul Hancock. I'm a postdoc researcher here at Curtin University. I do a lot of research into radio astronomy. I have a lot of interest in the evolution of the universe, exploding stars, galaxies that are colliding together, and pretty much anything that goes bump in the night.
What were your motivations to get into this field? I mean, some of them sound clear when you start saying bump in the night and exploding stars. It's pretty encouraging as it is.
Yeah, astronomy is one of those things where it's just really cool. And so when I was a little kid in Tasmania, we lived out in the sticks, out in bush behind a mountain. Didn't get hardly any TV signals or anything like that, and it was just super clear. Like nobody else lived anywhere around. Skies were magnificently clear. And I can remember this one night we went outside and there was the Aurora Australis. And I was a little kid and I was losing my mind, like, what is this? This is so cool. And my dad's like, oh yeah, it's the Southern Lights. It's pretty cool, you know. If you're interested, I got this book. Here you go, have a book about space. And it's this like monster tome about space. Everything from, you know, ancient ideas about how the solar system works to the moon landing to, you know, what the, you know, at the time the current state of astronomy. And from then on I was absolutely hooked. And that was probably, I don't know, I would have been in like grade 2, grade 3, something like that. So when I say read the book, I mean looked at the pictures.
Looked at the pretty pictures.
Yeah, mostly looked at the pictures.
I do that but now still. Some pretty pictures out there for creationists.
But ever since then, I've always had an interest in astronomy and in math. And so, you know, through school, I was always choosing science and math subjects. Got to university. I went to Sydney University specifically to study astronomy at the department there. And I was just, always been hooked. It's always my passion. And it's the sort of thing where you wake up in the morning and you're like, oh, it's Monday. That's cool. I'm doing astronomy today. So I guess I'll still go to work.
So what are you currently looking into? What's your research project at the moment? I've seen this funky looking antennae that I'll stick up on the website.
Yeah, so these little dipole things that we affectionately call spiders, we combine a whole heap of these together to make a radio telescope called the Murchison Wide Field Array. And I'm one of the many people who work on some of the science projects that aim to image the sky and then to see what's changing in the sky each night. So one of the projects I'm leading at the moment is actually to look at scintillation. So scintillation is probably familiar with a process where you go outside at night and some of the stars might be twinkling.
Yeah.
But in fact, as we all know, stars don't twinkle. The stars twinkle, but the planets don't twinkle. Just based on the fact that the planets are larger in the sky.
Yeah.
So that twinkling process also happens in the radio. But it's not our atmosphere that's causing it, it's actually our ionosphere. So our ionosphere can cause the twinkling. The gas within our Milky Way, the gas that lives between the stars, can also cause that twinkling effect.
So that twinkling effect caused visually is by our atmosphere and the way the light kind of refracts through.
Yes.
And then the radio signals are kind of refracted similarly in the ionosphere.
So instead of having dense pockets of gas in the atmosphere, which affects the visual signals, you've got different densities of the plasma in our plasmasphere or in the plasma, the gas between the stars.
So you said that you're looking into changes that could occur at night, and this is what the array's for. Could you tell us what are the applications or what the significance of this knowledge could bring?
So one of them that I'm trying to understand is if I can know how these galaxies are twinkling at night, that will tell me how the gas in our Milky Way is moving. Mm-hmm. And if we can have an idea about how the gas is moving, we can understand how that relates to forming stars. So star formation is linked to the turbulence in the gas. The more turbulent the gas, the harder it is to form the stars, but the more turbulence you have, the easier it is to mix all of the different elements that come from one star to mix it into other gas clouds. So
it's almost like settlement at the bottom of the tank. Like the more turbulent it is, the harder it is to settle and
exactly.
Substrate.
Yep. No, that's a great example. I like that one. So so understanding the turbulence and how it changes through our galaxy tells us how easy it is for stars to form in different parts of the galaxy.
You should sell yourself as fluid mechanics of space. That sounds very, very neat.
No, but then I have to do fluid mechanics.
True. So finally, what brings you here to RezBaz?
So a lot of the research that I do, so I said I love astronomy. The other thing that I love is computing. So I love all of the software that you need to use and needs to be developed to support astronomy. Astronomy is very much a do-it-yourself kind of thing. There are no commercial products and people making tons and tons of money out of astronomy. So if you want a piece of software that will turn these voltages from the telescopes into fancy images, somebody has to make those pieces of software. And unfortunately, the way that funding is usually given to universities and to research groups, it's for building things and for doing science, but not so much for developing software and providing all of those sort of support structures. So I'm really passionate about providing those support structures, and I see RezBaz as an opportunity to help other people get the software skills that they need to be able to do astronomy or agriculture or whatever it is that they want to do in science. And even outside of science, all of these skills are the kind of skills that people would need to go into whatever industry and whatever job and be the genuine scientist, physicist, which is a useful problem solver.
It is.
So
awesome. Thank you very much.
Cheers.
And that's it for our show today, guys. Thank you very much for joining us for this special episode of the 4 interviews at the Research Bazaar conference. Thank you very much again, Curtin, for allowing me to come along to this. And I'd also like to thank Ace Podcast for allowing me to be part of their network. They're a fantastic bunch of guys, and I strongly encourage you to contact them if you yourself have an interest in podcasting and would like to produce a show, or even just check out their website of acepodcastnetwork.com to find out what other shows they have on there. And don't forget to check out Vaguely Accurate's website at vaguelyaccurate.com to find out what else we have on offer. Sign up to our email list so you don't miss out on things, and check us out on Twitter at Vaguely Accurate without an E. Alright guys, thank you very much for your time today, and see you next time. Take care.